Bismillahir Rahmanir Rahim. Welcome again to this nice video. We are going to talk today about why it's dangerous to have our DNA or our chromatin or our genetic material outside the cell. So I'm going to talk about the extracellular activities of chromatin. Chromatin means a mixture of DNA and proteins, I mean histones. My reference today is this nice paper that was published in Cell Death and Differentiation in 2023, and its title is Moonlighting Chromatin When DNA Escapes Nuclear Control. And actually, I will cover the information that came in this paper in two videos. As we know, the usual ordinary place of our DNA or chromatin is inside the nucleus, under the nuclear control. But when this DNA or chromatin go outside the cell extracellular, it was found that it plays a significant role in driving the pathological background of many diseases. One example of DNA going outside is in the process of netosis or neutrophil extracellular traps that I was describing in other videos. In summary, why this extracellular DNA is dangerous? DNA serves multiple functions outside the cell, including triggering the interferon system, functioning as an autoantigen, and serving as a structural gathering for innate immune system proteins in the extracellular environment. When this extruded chromatin is not adequately cleared following the release of DNA from the nucleus into the extracellular environment, it can undertake a hidden role by participating in immune-inflammatory and occlusive disorders. In the nucleus, DNA plays an important role as a fundamental hereditary molecule, containing encoded information for gene structure and regulation, and the DNA in the nucleus is organized and bound to histones forming nucleosomes, and collectively these nucleosomes is known as chromatin. DNA, once extracellular, has the ability to expand in 3 dimensions and present additional functional activities that contribute to the promotion of inflammation and thrombosis. This phenomenon known as moonlighting refers to an additional and often hidden role or occupation. So while DNA primarily serves as a key player in heredity and gene regulation within the nucleus, its principal function in the extracellular space, whether in tissue or blood, is associated with immunity. The extracellular functions of DNA primarily arise from the charged phosphodiester backbone and its extended polymeric structure. The specific sequences and backbone of DNA play an important role in facilitating protein binding and forming the foundation for various intermolecular interactions. The translocation of DNA from the intracellular to the extracellular environment is the key mechanism that leads to the wide range of biological activities associated with DNA outside the cell. The release of DNA outside the cell can happen due to various factors such as cell death, stress, and injury. And among these, cell death is the primary source of extracellular DNA. When the cells die, DNA is generated as a byproduct and is typically regarded as debris. This extracellular DNA is supposed to be eliminated quickly or cleared. When DNA persists and reaches elevated levels outside the cell, it can acquire harmful properties and can be perceived as dangerous, as it has the potential to enter cells and engage with nucleic acid sensors. These sensors are integral to an internal host defense system that can be activated by foreign DNA originating from bacteria or viruses, as well as self DNA resulting from cellular stress or impaired nuclear activity. Apart from the unintentional or programmed cell death, the presence of extracellular DNA and chromatin can also arise through the process of neutrophil extracellular trap or NETosis that I described before in other videos. Net formation actually is a complex process formed by polymorph nuclear granulocytes, where the DNA is transported from the nucleus to the cytoplasm, and within the cytoplasm it combines with granule contents to generate nets, which serve important function in inflammation. The main constituents of nets, DNA, and histones are ancient and can be traced back to their presence even in archaea. Extracellular DNA, histones, and other antimicrobial proteins is also observed in various invertebrates like crabs, as well as in fish, birds, and plants. In mammals, extracellular DNA traps are not only produced by neutrophils but also by various other immune cells such as eosinophils, dendritic cells, monocytes, macrophages, mast cells, basophils, T cells, and B cells. Furthermore, DNA traps can also originate from non-immune cells such as endothelial cells, platelets, and cardiomyocytes. And we have to notice that the molecular characteristics of extracellular DNA or chromatin, as well as the composition of associated macromolecules such as histones and enzymes, vary depending on whether they originate from cell death or net formation. The translocation of DNA into the bloodstream is dependent on macrophages and can be affected by glucocorticoids and sex hormones. The size of extracellular DNA undergoes alterations when released during net formation. For example, DNA can have a high molecular weight ranging from thousands to tens of thousands of bases. However, in the bloodstream, the majority of DNA present is less than 200 bases in length. And apart from its soluble form, the extracellular DNA can also exist in a particle state. This particulate form of DNA is found within microparticles that are released from cells during apoptosis and are likely associated with membrane flaps on the cell surfaces. This particulate form of DNA is accessible and antigenically active and can be bound by monoclonal anti-DNA antibodies as well as by the sera from patients with systemic lupus erythematosus, resulting in the formation of large immune complexes. Additionally, mitochondria serve as another source of extracellular DNA in particle form, which is capable also of binding to anti-DNA antibodies. Extracellular DNA and chromatin have the ability to serve as a structural framework that organizes and enhances the activity of other mediators, thereby amplifying inflammation and promoting thrombosis. As regards the details of the process of net formation and degradation, I will not go into details in this video as I already described it in other videos. But we can say in short that NETosis have two main types, suicidal NETosis and vital NETosis. In suicidal NETosis, the cell will die, while in vital one, the cell will keep its viability. In suicidal NETosis, we have chromatin decondensation, then rupture of the nuclear membrane, and then this genetic or DNA material will be covered with the granules contents, and then the plasma membrane will rupture, and this chromatin covered by these enzymes from the granules will go outside the cells. While in vital NETosis, we have nuclear flipping, and we have DNA inside vesicles or flaps, and this DNA could come from the nucleus or from the mitochondria, and then it will be extruded through the cell membrane. Because of the wide range of signaling pathways involved in net formation, it's a challenge in identifying specific targets for therapeutic interventions in net-related diseases. And to effectively regulate net formation, there is a strong need for further research and development. The breakdown of nets by circulating DNases that I mentioned in the previous videos about netosis results in the release of net degradation products such as cell-free DNA, which is complexed with myeloperoxidase or neutrophilic elastase and histones. These nets degradation products exhibit toxic effects including complement fixation by cell-free DNA, induction of oxidative tissue damage by myeloperoxidase, or promoting thrombosis through localized proteolysis of tissue factor pathway inhibitor and activation of platelets, as well as cytotoxicity towards epithelial cells. It was identified that a total of 251 major cellular proteins in various compartments that make up the composition of nets. This includes important components such as azurophilic granule proteins, neutrophilic elastase, and myeloperoxidase, along with BAT4. BAT4, as I described and explained in other videos, is an important enzyme in the process of netosis. This enzyme is responsible for catalyzing the deamination of arginine to citrulline in the process called histone citrullination and facilitating chromatin decondensation. Among these proteins, only 74 were consistently detected in all nets, while others varied depending on the specific inducer of net formation. In this diagram, we can see the explanation of the process. So we start here by the presence of an event or skin wound. This will attract neutrophil to the site of injury or pathogen entrance, and these neutrophils will form netosis and extrude neutrophil extracellular traps, and then this will happen in just hours. Then macrophages and monocytes will secrete chemoattractant that will attract many of these neutrophils that will make aggNETs or aggregates of nets, and these aggregates will help to resolute the inflammation by degradation of the inflammatory products. And then after doing its function, DNase, the one that destruct the nets, will work on these nets to make nets degradation products, and then macrophages come and remove all debris and make clearance of the area. So this is a normal condition, while if we have a problem with this clearance, we have reduced clearance, then the fibrin will be attached to the nets and make aggregates, and this fibrin will be citrullinated and resist plasmine lysis, and also the aggregates of nets with this combination with fibrin will be resistant to DNase, and so this condensation of nets with fibrin will be the stimulus for chronic inflammation and other problems like stones and tophi. So at the site of injury, the binding of neutrophilic elastase and other antimicrobial proteins to the extruded chromatin of nets mediate the digestion and elimination of microbial pathogens. And at these areas, because of the presence of high density of neutrophils, neutrophils tend to aggregate and form enzymatically stable clumps that is called aggregated nets or aggNETs. Enzymatically stable clumps known as aggregated nets or aggNETs play an important role in immobilization, neutralization, and potentially eliminating bacteria, fungi, viruses, parasites, and they effectively prevent the dissemination of these pathogens. Also, aggNETs can contribute to the resolution of inflammation. The chromatin fibers of aggNETs are attached with numerous cytoplasmic and granular proteases As a result, aggNETs have the ability not only to scavenge inflammatory mediators but also to degrade these molecules. This proteolytic activity is particularly important in the breakdown of highly cationic histones that may have entered the extracellular space during net formation, so safeguarding surrounding tissues from chronic damage and facilitating the resolution of tissue homeostasis. aggNETs also serve to sequester and degrade inflammatory cytokines and chemokines. When there is a disruption in the aggregation and clearance of NETs, it can contribute to the onset of autoimmunity and have hazardous effects. Consequently, an unbalanced immune response and an apparent relationship between net formation and degradation can give rise to severe diseases. I hope that you have enjoyed the first video about the dangerous effect of extracellular chromatin, and I'm reminding you again to subscribe to the channel so that you can get any new video, and hoping to see you soon in the second video about detailed information for the role of extracellular chromatin in many debilitating diseases. Thank you.